Alright, we'll be taking a look at lymphatic organs and also lymphatic tissues during this discussion. And I want to first start out by giving you some definitions to help you through this discussion. The first will be the definition of what a lymphatic organ and also a lymphatic tissue is. Lymphatic organs, by definition, contain a connective tissue covering called a capsule. So quite simply put, this connective tissue covering, a capsule, is something that will be present in all of your lymphatic organs. By contrast, lymphatic tissues do not have a capsule. So that would be the difference between the two. Organs have a capsule, tissues do not. Now, for the purposes of your quiz and test, you will need to know examples of each. Examples of lymphatic organs include the thymus, lymph nodes, spleen, and also red bone marrow. When we consider the tissues include tonsils, something found in the small intestine referred to as Peyer's patches, the appendix, and also lymphatic nodules. So we'll be taking a look at some of these as we go through this discussion. Next, I want to give you another classification or another way to classify these lymphatic organs and tissues, and that is based upon their function. Based upon their function, we can classify the lymphatic organs and tissues as either primary or secondary. Now, you should keep in mind what primary and also secondary refer to. Primary lymphatic organs are sites where immune cells become immunocompetent. Immunocompetent just means that this is where these immune cells will mature. Now, the immune cells that we're talking about are actually lymphocytes, and there's going to be two locations where they will ultimately mature or become immunocompetent. That would be the thymus and red bone marrow. So there are only two primary lymphatic organs, thymus and also red bone marrow. By contrast, we also have secondary lymphatic organs and tissues. Secondary lymphatic organs and tissues are not sites where immune cells become immunocompetent. They are sites actually where immune responses occur. So secondary lymphatic organs and tissues are sites where immune responses occur. Primary lymphatic organs are sites where immune cells become immunocompetent. Now we'll take a look at several of these organs and tissues throughout the remaining discussion, and you should remember the classification for each, and I'll have a few words to say about each as we go through them. So here we have the thymus, which is a primary lymphatic organ. The thymus is going to be the site where T cells mature. So when you hear T lymphocytes, it's actually implying that the thymus is where those cells mature. Grossly, the thymus is located within the mediastinum. It's situated directly on top of the heart. It is a bilobed organ, meaning that it has two lobes. If we begin to take a look at the thymus histologically, you'll see that, like all organs, it does have a capsule. Now, the pattern that we see arise with any lymphatic organ is that its capsule typically will invaginate or extend into the actual tissue of that organ. These extensions of the capsule into that organ are called trabeculae. So you see the trabeculae throughout the diagram also. These trabeculae separate each lobe of the thymus into smaller units called lobules. And each lobule contains its own cortex, noted here in the darker purple region, as well as its own medulla, located centrally typically, and depicted also in the lighter purple region. Randomly, you will see the appearance of what are called thymic or Hassel's corpuscles within the medulla of the lobule, and these are unknown collections of cells. We really don't know what they do. So that's the anatomical breakdown there of the thymus. Now, as I said before, it is a primary lymphatic organ, and it is a site where T cells will ultimately mature. As far as the thymus is concerned, over the course of our life, it progressively will become smaller and smaller. If we look at the previous diagram here, you see it says that this is the thymus of an adolescent. It's fairly large in size during adolescence, but it becomes progressively smaller throughout our life because of a property of our immune system called memory. It's thought that as we get older and as we're exposed to more antigens, we no longer need to train as many new T cells to try and fight off the foreign antigens once we encounter them. So because we, in theory at least, need fewer and fewer T cells as we get older, the thymus begins to get smaller and smaller. So, as you see here, like I said, the thymus of an adolescent is fairly large, but as we get older, it should decrease in size. Also, the T cells that are mature within the thymus, many of them do not ultimately reach full maturity. This process of gaining immunocompetence is just that, a process, and not every T cell that begins that process will make it. In fact, the overwhelming majority of those cells don't go through the entire process. So, very few T cells are able to make it through training, quote unquote, there, to reach full immunocompetence. I want to next look at the next organ in the list. This is a lymph node, which is a secondary lymphatic organ. And the lymph nodes ultimately, if you look at them grossly, are going to be clustered together in various groups located throughout the body. I'm going to go back to a diagram I usually refer to as the lymphatic lady. And you can see when we cluster these nodes together, they get named upon the anatomical location that they're located within. So you have, for instance, submandibular nodes beneath the mandible, cervical nodes within the neck, axillary nodes within the armpit, iliac nodes along the distribution of the common iliac arteries, and inguinal nodes in the hip area. So the nodes simply are clustered together, as I said, and they're named for the area of the body that they're clustered within. Now the main job of these lymph nodes will be to filter lymph. They filter lymph in an effort to detect the presence of foreign antigens. And should they ultimately detect those foreign antigens, then the lymph nodes should respond to those antigens. You'll see within lymph nodes collections of cells involved in immune processes. These include the B cells, the activated form of a B cell referred to as a plasma cell, macrophages and dendritic cells, which are collectively what we refer to as antigen presenting cells, or APCs for short. We'll talk about those later in this chapter. And also T cells. So all the cells necessary to begin or initiate an immune response are located here within the lymph node. Now in the bottom right hand corner you'll see a little flow pattern that you definitely need to remember for the purposes of your test. This describes the flow of lymph through a lymph node. Lymph is delivered to the lymph node via what are called afferent lymphatic vessels. When you see that term afferent, think add, because that is what these lymphatic vessels do. They add lymph to a lymph node. From the afferent lymphatic vessels, lymph then flows through a small opening just beneath the surface of the capsule here. This is called the subcapsular sinus. And you can see it in random locations throughout the lymph node just below the capsule. However, they only tag it up in the top right hand corner. From the subcapsular sinus, then lymph would flow into what we call trabecular sinuses. As I said a few moments ago with the thymus, any lymphatic organ, its capsule ultimately will extend into the tissue of that organ and form these trabeculae. So you see the solid white lines throughout here. These are going to be your trabeculae. Around the trabeculae would be the trabecular sinuses, and that's the point at which we're going to see lymph be released from the sinuses and enter the outer portion of the lymph node called the cortex. And this is where all those immune cells are going to be located. The T cells, the B cells, the dendritic cells, the macrophages. You see these larger collections of B cells and antigen presenting cells called germinal centers here. So these germinal centers located throughout the cortex along with other cell types. And the lymph at that point will then be filtered. It'll be screened for the presence of any foreign antigens, and should those B or T cells recognize a foreign antigen, they should respond to it by initiating an immune response. But at the end of the day, that lymph is meant to flow. And it needs to continue to flow throughout that lymph node and ultimately exit the lymph node. So the lymph then moves toward the central area of the lymph node, which is its medulla. And in the medulla you'll find the medullary sinuses. The medullary sinuses begin the drainage of this lymph, because from the medullary sinuses, lymph then will drain toward the exit point, or the door, if you want to put it that way, of the lymph node, which is called the hilum. And emerging from the hilum will be the final in the pathway, the final structures in the pathway, that is, efferent lymphatic vessels, which will allow lymph to exit that lymph node. So, as I said at the beginning of that, please definitely remember that route of lymph through a lymph node is down there in that lower right hand corner for you in that box. It's something that you will see come up again, and you should keep in mind that these lymph nodes are secondary lymphatic organs, and that they're going to be involved in filtering lymph, and they contain these collections of B cells, T cells, and antigen presenting cells collectively. The final organ we want to take a look at here is the spleen. The spleen is located in the left upper quadrant of the abdomen. It's a fairly flat bean-like structure, and it has three main functions. The first function is to destroy old red blood cells. Secondly, it stores platelets. And thirdly, it participates in immune responses. So please keep in mind those three functions: destroying old red blood cells, storing platelets, and also participating in immune responses. Now, if you look at the spleen histologically, you'll see it also has a capsule, so it is a secondary organ, like we said a few moments ago. That capsule forms extensions throughout the tissue called trabeculae, which you see here. And then you'll see two different types of tissue collections within the spleen. They are called red pulp and also white pulp. The pulps are named for the majority cell type that's found within them. Red pulp has mainly red blood cells, but there are other cells that are there as well. And white pulp, mainly white blood cells. The trick of this is that the white pulp actually looks darker, and the red pulp actually looks lighter, and it's due to the difference in staining because of the presence of more nuclei within the white pulp. You'll see usually white pulp clustered around small vessels called central arteries. These central arteries are delivering blood, obviously, to the spleen, and it receives a good amount of blood relative to its size, remember, because it helps destroy old red blood cells, so it ultimately must filter those red blood cells from blood, and in order to do so, it would need to receive blood in the first place. So, once again, the spleen is a secondary lymphatic organ with the three major functions that I mentioned to you before. Keep in mind also the different tissue types found, red pulp versus white pulp, and you should also keep in mind that it is a secondary organ, and that should be sufficient. Now, to kind of start to wrap up the discussion about lymphatic organs and tissues, we need to discuss the tissues. Many of these tissues fall up underneath the category of lymphatic nodules. And many of these lymphatic nodules are actually going to share some common characteristics. First of all, many of them are located within the mucous membranes of our body. These mucous membranes line the cavities of the body that are exposed to the outside of the body, so you'll see these mucous membranes associated with, for instance, the digestive system, respiratory system, urinary system, and also reproductive system. Now, in addition to being located in the mucous membranes, many of these lymphatic nodules also are fairly small in size. They're actually usually solitary or isolated, meaning that they occur or you see them individually or by themselves. And they're actually also random. And by random, I mean there's usually no consistent pattern to where they're located. If you went 2 inches into the stomach of one individual, you may see some of these lymphatic nodules. If you went 2 inches into the stomach of another individual, you may not see any lymphatic nodules. Now, as long as they fit that criteria, these lymphatic nodules are usually just referred to as lymphatic nodules. Or sometimes we use the acronym MALT, which stands for mucosa-associated lymphatic or lymphoid tissue, because it's lymphatic tissue associated with mucous membranes. However, if those characteristics are not followed, then usually what happens is the lymphatic tissue gets a name. That's what happens in the case of, for instance, the tonsils. They are secondary lymphatic tissues, but there's consistency between people with tonsils. Everybody has the same tonsils as long as you have not had yours removed. Secondary lymphatic tissues would also include the appendix, which, once again, is no longer random. It's no longer isolated because you'll see it from person to person. And we could also include structures found within the small intestines called Peyer's patches. Peyer's patches are no longer random. There's consistency, and you'll find them from individual to individual. So when you lose those characteristics of being random, isolated, and also, in some cases, small, the lymphatic tissues actually get a proper name, such as tonsils, appendix, and also Peyer's patches. So that's our discussion about lymphatic organs versus tissues. Keep in mind those points that we made, and you should be straight as far as the test is concerned.